Hi guys, this is Mrs. White coming to you with the 11.1 work of Gregor Mendel lesson overview. So this section really talks about what Gregor Mendel did for the field of genetics and how he accomplished it. It also gives a basic introduction to both Punnett squares and to genetics with dominance and recessive traits. So let's get started. So how does an organism get its unique traits? Well, they inherit them from one parental generation to the next. So half of your genetics come from your mother and half from your father. And it's this study of inheritance that is really genetics. So every living thing inherits genetic material from a parent or parents. This delivery of characteristics from parent to offspring is called heredity. The scientific study of heredity is known as genetics, which is a key to understanding what makes us all so unique. So Gregor Mendel was an Austrian monk. So back in the day, if you wanted to study things, your best bet was to become a monk. So you had plenty of free time to study both theology and the natural world. So that's what Gregor Mendel did. And he spent most of his time growing pea plants. So peas work really well, partly because they're small and easy to grow, and because a single pea plant can produce hundreds of offspring. They can also produce up to 2 rounds of offspring in a single growing season. It is what we call a model system. This means that the information we get from this system can be applied to more complex organisms with longer life cycles. So they're very convenient and they can tell us how our bodies will actually function by looking at these simple organisms. If we had done these experiments with ourselves, human beings, instead of pea plants, it would have taken centuries to do, whereas it only took years, decades for Gregor Mendel. So let's take a look at the flower. There's a male part and a female part. The male parts contain pollen. The female parts contain the eggs. So what Mendel did was when he wanted to interbreed the plants, he would remove the male parts.
Are you making a video?
This is my son, James.
Hello.
And yes, I am making a video. So what Gregor Mendel would do... Sorry for the interruption, guys. So what Gregor Mendel would do, he would remove the male parts from the plant, and then he would take the pollen with a small paintbrush, and he would fertilize whichever plant he wanted to. The thing with pea plants is they can breed asexually, so they can self-fertilize.
Fertilize? What's fertilize?
So during sexual reproduction, male and female reproductive cells join in a process known as fertilization to form a new cell. It produces a tiny embryo encased within a seed. So pea plants are normally self-fertilizing, which means that the sperm cells fertilize the egg cells from the same flower. A plant grown from a single seed produced by self-pollination inherits all of the characteristics from the single parent. So basically it has one parent instead of two. So what Gregor Mendel did was he had a stock of true breeding, meaning that they were self-pollinating and would produce offspring with identical traits to themselves. In other words, the traits of each successive generation would be the same. A trait is a specific characteristic, such as seed color or plant height, and it varies from individual to individual. So this is what I was talking about with Gregor Mendel using the paintbrushes to go ahead and move the pollen to whichever plant he wanted. What he did was he crossed his true breeding plants to produce a hybrid. He did this by, like we discussed, cutting away the male parts and then crossing them. This cross-pollination has 2 different parents. Thus, they inherit traits from both of the parents, and he would then study the results. So he studied several different characteristics, including seed color, height, seed shape, and pod shape. There were actually 7 contrasting characteristics that he studied, and the crosses between parents with different traits are known as hybrids. So here's a picture of all of the different hybrids that he crossed. The original cross of the true breeding pair is known as the P, or the parental generation. The first filial generation is the generation that comes from the parents being bred together. As you'll notice, only the dominant traits are seen in the first filial generation. That is because the dominant traits are masking the recessive traits, but bear in mind the recessive traits are still there. So for each trait Mendel studied, they had the characteristics of only one of the parents. So it looks like the recessive one disappeared, but it is still there. So he drew two conclusions. His first conclusion was what led to our current understanding of inheritance.
Can I just say one word?
An individual's characteristics are determined by factors that are passed from one generation to the next. Scientists now call these factors that are passed from one parent to the offspring as genes. Each of the traits Mendel studies is controlled by one gene that has a dominant and a recessive form. The different forms of these genes are called alleles. Mendel's second conclusion was the principle of dominance, which states that some alleles are dominant while others are recessive. Any organism with at least one dominant allele for a particular form of a trait will exhibit the form of the trait. An organism with a recessive allele for a particular form of a trait will exhibit that only when the dominant allele is not present. In Mendel's experiments, the allele for tall plants was dominant and the allele for short was recessive. So as you can see, the allele for yellow seeds is also dominant over the recessive allele for green seeds. So how are the genes distributed to offspring? That's where the principle of segregation comes into play. The allele for each gene segregate from each other so that each gamete or sex cell carries only one allele for each gene. So as you can see, the first generation has a tall true breeding plant being bred with a short true breeding plant, which produces 100% tall plants, but they will all be heterozygotes, which means that they have two different alleles. The parents were homozygous because they only had one type of allele. And then in the F2 generation, you have 3 tall plants and 1 short plants. You have 2 homozygous, 1 big T, 1 little t, and 2 heterozygotes, big T, little t. So segregation occurs when the alleles separate during the meiosis stage, which is something we'll learn about in the next couple lectures. So each sex cell gets one allele. So let's assume that each F1 plant, all of which were tall, inherited an allele for tallness from the tall parent and an allele for shortness from the short parent. So you can see all the big T's and the little t's. So each of them segregates, and they can assort randomly with any other allele from the other parent, producing our big T, big T, little t's, and little t's. So 2 homozygous sets and 2 heterozygous. All right, thank you for watching. Next video, we'll be talking more about Mendel and his work, and we'll be getting more into Punnett squares and different parts of genetics. Make sure to tune in, like, and subscribe. See you later.